Module 9, Reproductive Technologies and Their Role in Livestock. Genetic Improvement. Module. Overview. Module 9 delves into the advanced reproductive technologies, ARTS, used in livestock breeding, highlighting their impact on genetic improvement, productivity, and breed conservation. This module provides a comprehensive understanding of artificial reproductive methods, molecular genetics integration, and strategic application to maximize genetic gain and herd performance. The focus is on reproductive physiology, estrus synchronization, artificial insemination, embryo transfer, in vitro fertilization, IVF, cryopreservation, and cloning, as well as ethical, economic, and practical considerations in implementing these technologies in modern livestock production systems. The module emphasizes the synergy between conventional breeding strategies and ARTS, highlighting how they accelerate genetic improvement while reducing generation intervals and increasing selection accuracy. Key topics in reproductive technologies. Fundamentals of reproductive physiology in livestock, anatomy, and physiology of male and female reproductive systems. Estrus cycles in different livestock species, cattle, sheep, goats, pigs, poultry. Hormonal regulation of reproduction, including roles of FSH, LH, estrogen, and progesterone. Fertility challenges and reproductive disorders affecting genetic improvement. Estrus synchronization and ovulation control use of hormonal protocols, prostaglandins, GnRH, progesterone devices, to synchronize estrus. Benefits for planned breeding, fixed-time artificial insemination, FTAI, and herd management. Strategies to enhance reproductive efficiency and optimize breeding schedules. Artificial insemination, AI, principles, techniques, and advantages of AI in livestock. Semen collection, evaluation, processing, and storage. Timing of insemination and AI protocols for different species. Economic and genetic advantages. Widespread use of superior sires, increased genetic diversity, reduced disease transmission. Embryo transfer, ET, technology superovulation, and embryo collection techniques. Embryo grading, preservation, and transfer methods. Applications. Rapid multiplication of superior genetics. Conservation of elite or endangered breeds. Limitations and technical challenges. Cost, expertise, and reproductive response variability. In vitro fertilization, IVF, and oocyte manipulation. Oocyte recovery, fertilization, and culture techniques. Use of IVF to propagate elite genetics and produce multiple offspring per donor. Integration with cryopreservation and sex sorting technologies for targeted genetic improvement. Cryopreservation and genetic resource banking. Freezing of semen, embryos, and oocytes for long-term storage. Advantages: preservation of rare breeds, disease-free genetic materials, and strategic breeding programs. Techniques: slow freezing, vitrification, and liquid nitrogen storage. Cloning and advanced molecular techniques. Somatic cell nuclear transfer, SCNT, and its applications in livestock production. Benefits for reproducing genetically superior animals and conservation. Ethical, regulatory, and economic considerations in cloning. Integration with genetic evaluation and selection programs. ARTs as tools to shorten generation intervals and amplify selection response. Combining reproductive technologies with genomic selection, marker-assisted selection, MAS, and breeding indices. Case studies: dairy cattle, goats, sheep, and pigs. Challenges, risks, and ethical considerations. Technical limitations, efficiency, skill requirements, cost. Health risks, reproductive disorders, reduced fertility in donors. Ethical issues, animal welfare, public perception, and regulatory compliance. Practical applications use AI and ET to rapidly multiply genetically superior sires and dams. Implement estrus synchronization to maximize herd reproductive efficiency. Apply IVF and embryo transfer in high-value genetic programs. Preserve endangered or elite breeds using cryopreservation techniques. Integrate reproductive technologies with genomic selection programs for accelerated genetic gain.